Hello, and welcome to ISPE's I Speak podcast, the place you can come to learn about hot topics, news, and trends from experts in the global pharmaceutical and biopharmaceutical manufacturing industry. My name is Barbara Peck, and I'm the Manager of Community and Industry Recognition with ISPE, and I'll be your host today as we talk with our panelists about pharmaceutical water, specifically water for injection using non-distillative methods, the ISPE DACH approach. This podcast episode is a follow-up to our very successful webinar series. Actually, our very first webinar episode from April 2019, Fritz Roeder was the presenter for the session, and he has assembled a panel of experts today to answer some viewer questions from the session and to explore the topic a bit more in detail. Thank you for joining us today, Fritz.
Thank you, Barbara. I'm happy to be here, and also let me thank ISPE to make this webinar able to be held. This was really great, and I was so overwhelmed about the number of registrations we had. And yeah, and in addition, this webinar was for free. Perhaps this was also a reason why we had so many registrations, and also in the webinars after mine. And this is a great opportunity for our members to gain more knowledge and to get more experience.
It's been very successful with this webinar series. I've actually had 8 sessions at this point, so you kind of set the stage for getting a lot of registrations and getting some interest around this. And I think it actually kind of worked out perfectly that we had this webinar session. We were internally discussing here at ISPE about having a webinar program, and at the exact same time, I happened to see a post from you on the community asking if anyone would be interested in listening to a webinar. So it was the stars were aligned. It was a perfect timing. So I reached out to you. We had our first webinar, and the rest is history. So thank you for being our first webinar guinea pig, and then now for doing this podcast to go back and answer the many questions that we received from that webinar. So thank you. Okay, so with that, I thought we should just get started. I'll do just a quick intro of our panel, and then we'll do a quick overview of what the webinar was for anyone who missed it, and then we'll get started in the Q&A. Does that sound good?
Yes.
Okay, so today's panelists are Fritz Roeder from Merck, who's based in Darmstadt, Germany, and is an experienced project manager and engineer with broad knowledge in the pharmaceutical industry. He is an experienced speaker and author on a variety of pharmaceutical water topics. And then our next panelist is Michael Cleaver of Roche, and he's based in Pennsburg, Germany, as an expert in the field of clean media operations with a strong engineering background in utility plants. He did a lot of research in the field on ultrafiltration and rouging. And finally, we have Jochen Schmidt. Jochen has been working in the pharmaceutical water sector for more than 20 years. He's currently the process utilities lead at Pharma and Biotech Engineering Service Provider, CRB, based in Basel. His main focus is on the conception, process selection, and dimensioning of complete water systems for the generation, storage, and distribution of pure water, water for injection, and pure steam. So all of our panelists are heavily involved in the DACH water and steam group. So I just want to welcome you, and we can get started. Thank you, everyone. Okay. Fritz, would you like to give just a quick overview of what we talked about on the webinar? I know it was a few months ago, but for anyone who missed it, that way they don't feel like they're missing anything.
Yeah, so I mean, the main reason for this webinar was many discussions of the ISPE DACH group concerning cold water for injection or water for injection using other methods than distillation, because as many people know, since April 2017, European Pharmacopoeia allows to produce WFI using other methods. And meanwhile, the additional water quality highly purified water has been removed in April 2019. And so this means there's a huge change in the regulatory situation. And so this ISPE DACH Water and Steam Expert Group decided to write a handbook, which has already been published in the German language, to answer many questions that are open and also that were opened by the regulatory guidance. And yeah, so we decided also to offer that webinar to make some positions clear. And yeah, so basically this webinar covered many questions about the control strategy, about suitable technologies for the final treatment step of a cold WFI system, how to validate the final treatment step, how much control and how much oversight is needed, if there is more control needed compared to a traditional HPW system. We were discussing why we set up a new standard to determine the cutoff of an ultrafiltration module, because there is no standard existing yet, or the discussion about what to do with existing highly purified water systems where no monograph is anymore in place. And finally, a last topic was the topic requalification from HPW to WFI. If somebody decides to upgrade his system or requalify a system, what would be needed? So these were main aspects of the webinar in April, and all ISPE members are still allowed to watch the webinar. So if you want to watch it, feel free to register.
And we'll make sure to include a link for the webinar in the episode notes so that you'll just be able to click on the link. So thank you, Fritz, for that background. So I know we went through the questions that we received from the audience. So then just picked a selection of questions and we'll just jump in, and the three of you will answer them as best as you can. Okay, let's get started.
Yeah,
All right.
so
Sorry,
may
Barbara.
I?
Yes.
Sorry, Barbara, for chiming in just before
No,
we
go
start.
ahead. Go ahead.
We are talking about cold WFI system. I was corrected by my American colleagues that they are not talking about cold systems because their understanding of cold is maybe 5 to 8 degrees. They are talking in the US about ambient storage and storage and distribution systems. So when we speak about 20 to 25 degrees or 15 degrees system, we mean ambient. So, but we say cold, what Americans consider to be lower temperatures. That's just for clarification.
Oh, that's great. Thank you for clarifying.
May I also add that we had about 100 questions, and I want to thank all the users that had registered to the webinar and that are participating so greatly in the discussion. And so we picked more or less the 10 most interesting questions for you right now, and we'll have a discussion on this.
Okay. All right. So our first question is for Michael or Fritz. Moving towards HPW to hot or cold WFI, the upgradation requires some minor modification at a computerized system level. Do we need to validate it to its fullest, or will we be addressing those changes in the addendum?
Yeah, so, well, it depends on the level of change you have to implement on the system and your company's strategy behind that. If the right systems are already in place and no or only minor changes are required, so there's, from my perspective, no need to run an entire requalification process. If there's a major change in your system, it makes sense to requalify the plant.
Yeah, so what I can add from my side is it's all about the control strategy and also the data you have. So if you had run an existing system, have a look at the data you got. Did you have it in a good state of control, or did you have many deviations? And what's the entire control strategy like? What does it look like? Do you have to add maintenance tasks? Do you have to add the use of rapid methods maybe? So these are all the questions you must ask and decide how much requalification effort is needed.
Okay, thank you guys. Our next question is for Fritz. During the session, you mentioned that notice is given to the supervisory authority of the manufacturer before implementation. The viewer didn't understand your answer. Could you explain a bit more in detail of what this supervisor authority is?
So this sentence is written down in the WFI monograph 169 from the European Pharmacopoeia. So, and we were also asking some authorities, because I mean, if you're based in Europe, if your facility is based in Europe, it's easy because you just contact your local responsible authority. That's easy. But for the listeners in the USA, for example, or in India, because this question was from a person from India. Your responsible authority should be EMA, where you also get your GMP manufacturing license from. So it's dependent where your site is located, who's responsible for you.
Okay. Our next question is for Jochen. In spite of the EMA Q&A and the DACH guidance, many national regulatory agencies such as Anvisa, China, Russia just won't accept non-distillation techniques. Do you know why that is, or do you anticipate they will eventually accept non-distillation techniques?
Hello, before I start to answer, I would like to say thank you to be invited to this great event. Now let's go on to answer the question. According to my recent investigations, actually only the Chinese pharmacopoeia and the International Pharmacopoeia of the World Health Organization still insists on distillation as final treatment step for production of WFI. From what we know, India changed their WFI monograph accordingly in their last revision of the Indian Pharmacopoeia. It was published in end of 2018. Brazil and Russia allow alternative technologies since many years, but not many people know that. The future expectation is that the World Health Organization recently announced that they will adapt their WFI monograph in the next revision of the International Pharmacopoeia. From what we know about the development in China is that China is not going to adapt their WFI monograph in the next revision of the Chinese Pharmacopoeia, which will be published in 2020. We expect it in April. That means probably another 5 years restriction on distillation if you want to export your drug products to China. And with regard to the question why the development towards membrane technologies goes so slowly in the pharmaceutical industry, I can only answer tradition, and that the pharmaceutical industry is in principle a relatively hostile environment with regard to innovations. So we know since many years, I would say 20, 30 years, that the semiconductor industry, for example, is able to produce ultra pure water via membrane technologies, which has a far better quality than WFI. And this water is so good that bacterial contamination is no longer possible due to the very low nutrient content. So therefore, it's time to move towards the membrane technologies and also to reduce the limiting values because they are far too high, in my opinion.
Okay. Since we're talking about regulatory questions, I have another for Fritz. One of our viewers asked if it's mandatory to get FDA approval when upgrading to new RO unit from HPW to WFI. It's a lot of acronyms there.
Yeah, so no, basically it's not required to get an FDA approval because highly purified water was a water quality, compendial water quality that only existed in Europe. If you look at the US guidance, you can read in ISPE 1231 about high purity water, but that's something different. It's not the same like the European highly purified water was. So the FDA would never have approved a highly purified water, whatever system, because this quality didn't exist in US. And therefore, the FDA has nothing to do with it. So this is something you must clarify with the European authority. But if you were operating a purified water system, for example, and now want to upgrade it to WFI, this might be subject to changes with the authority.
Okay, thank you. So our next question can be answered by any of you guys. What are the typical costs of cold WFI versus distilled WFI?
Yeah, so let me start here. It really depends on the cost structure you apply. So for the operating costs of an ultrafiltration, it could be half of the cost for distillation if the same level of system control would be applied. But based on the requirements you find in the EMA Q&A document, the level of control for cold WFI system is now much higher. This is at least the expectation from EMA. That means the cost benefit of a cold WFI system or ambient WFI system shrinks down to approximately 30% based on my knowledge.
I can also add that there's a lot of discussion going on in the pharma industry concerning water costs, because people don't really compare the right things. But I guess in total, there's few data available about the total cost. So what do you include? Maintenance, deviations, calibration, not only media supply cost. Very interesting is sampling costs. And so my personal views, I share the opinion of an article from a German company in the Farm recently, where a cubic meter cold WFI or ambient, like Jochen says, costs in Germany about more or less 53 euro compared to 77 for a hot system. So this is data that is, for me, well, mostly reliable or plausible. So yeah, that sounds reasonable for me. And yeah, but it's so difficult to include the total costs, because you always must differentiate what's inside and what is not.
Yeah, I totally agree that it really depends on the structure you apply for the costs, and what's inside and what is outside of that evaluation. Yeah.
The scope, yeah.
I fully agree with you guys. Most important for an objective comparison is the consideration of the monitoring costs as well, and the standby losses especially. So for example, the continuous blowdown of a distillation in standby is normally not considered, because most of the operation cost comparisons are made just when you look at the operation of the system. Standby losses are not considered, and this could reach at almost the same value as the cost for during the production time. So I use, it differs a little bit from the value Fritz said before. 50 euro is including everything. I would say this is, if I remember, Fritz, monitoring cost was also included in this consideration.
Yeah,
It makes sense.
that's the highest cost to my mind, monitoring, sampling, incubation.
Absolutely. For pure operating costs, that means utility costs for the production, I normally use another rule of thumb, which I developed based on operating costs comparison I made by myself. My rule of thumb is that hot membrane-based WFI is about twice as expensive as ambient membrane-based WFI. That means the difference between 20 degrees and 80 degrees makes almost double the price, just because of the heating. And if you produce WFI via multi-effect distillation, hot WFI, this is approximately 3 times as expensive as ambient membrane-based WFI. So as an example, ambient membrane-based WFI may be 10 euro per meter cube, then hot membrane is about 20 hot membrane-based WFI, and if you use a distillation, it's about 30 euros per cubic meter. But you see, there are big differences. So every case has to be considered in detail based on their local values they have on site.
Okay, thank you. That was a good discussion. So for our next question is from Michael. This one was worded a little bit strangely. I think maybe, hopefully I'm getting the right question. So for our benchmark, the Q&A lets us consider a high amount of TOC measurement locations. Do we have any indications or suggestions?
Yeah, sure, based on broad experience, but in the beginning you have to do a proper raw water analysis. This is of utmost importance, and if based on this raw water analysis the right setup of the WFI plant is in place, it seems to be sufficient to measure the TOC at exactly 2 locations. One is the downstream of the RO, respective EDI, and one is downstream of the ultrafiltration, either in the feed flow of the loop or in the return flow.
So which is then meant as the final water quality, right?
Sure, yes, you're right.
Yep.
So I would like to add, guys, that it's often made a mistake that the TOC value is brought into a relation with the microbiological contamination. We have to state that via the TOC value, you cannot measure microbiological contamination. It just measures the total organic, the content of total organic carbon, which means the higher the TOC is, the higher the risk of microbiological contamination is. And with regard to the TOC measurements in the generation system, I agree completely. It makes only sense to install a TOC measurement after a treatment step, which normally reduces the TOC. So it makes no sense to install it after each treatment step when the step has no impact on the TOC.
And we wouldn't also be able to afford that because typically TOC measurements cost some money, as well as rapid microbiological methods like bioburden measurement devices. This is expensive, and we aren't able to install these at 7 locations like stated in the EMA Q&A or so. We must choose 1, maybe 2 locations and live with that. Maybe we can use one device with several sample inlets, but then you get the risk of impurity carryover and so on. So it's not such an easy topic. No? So we must focus on the most important locations.
Okay. Any other thoughts on that? Okay. So our next question is for Jochen. What is the max residual ozone concentration in WFI that will be in direct contact with product so as not to impact product quality?
Sorry, Barbara, I have to add a point towards the last question. I just forgot to switch on the mic.
No problem.
In principle, I have to say that more measurements in the system does not consequently makes the system better. So we should work on the function and improve the water quality. That has a much bigger effect on the quality of the product finally. So that's just a comment on this point with regard to a lot of TOC measurements. So coming to the point with regard to the ozone, basically we expect that no ozone should reach the production facilities. Oxidizing substances are like ozone. It's the strongest oxidizing substances in. Yeah, I don't know. I don't know a stronger substance like ozone is generally undesirable in water during production because it could damage both the product and, for example, bacteria who produce the product in biotechnology processes. To my knowledge, such a connection, that means product or bacteria destruction by ozone, has not yet been established or found everywhere, but only the potential risk is sufficient to get to the requirement that we need ozone-free water during the production. So the ozone depletion is in such a water system usually carried out in the loop supply line through a UV system, and it is monitored by an ozone measurement downstream of the UV. The weak point of this design is the detection limit of the ozone measurement, which is normally in an amount of approximately 5 ppb. This means even if the measurement shows 0 ppb or shows nothing, you cannot be sure that there is a residual amount of ozone traces in the water up to 5 ppb. So therefore, it is very important to correctly dimension and monitor your UV system. So if it's designed well and the function is controlled via a calibrated UV sensor, you can by physics ensure that ozone is removed completely.
Okay. Thank you for that. Our next question is for Michael. As filtration separation techniques employ higher pressure on the dirty side compared to the clean side, do you consider this risk element correctly covered by using the same monitoring instrumentation as for distillation techniques?
Yeah, so let me answer this question this way. So the instrumentation for pressure measurement should encompass feed flow, the concentrate flow, and the permeate flow. The return flow, of course, only in the ultrafiltration, if the ultrafiltration is operated in cross-flow mode. For dead-end systems, this doesn't really exist. And based on these three respective two measurements, the transmembrane pressure is calculated. This normally takes place in the DCS system. And the transmembrane pressure is the most significant indicator of the condition of the RO respective ultrafiltration. So you finally have to install a couple of pressure measurements to have good oversight of your system.
Yeah, and that's something we recommend in the handbook, in the WFI handbook. This is one aspect where we think we can have a lot more control for cheap money when measuring performance parameters of RO and UF. Yeah, this helps us a lot. And in comparison, therefore, we omit many locations for TOC measurement or online bioburden measurement, for example. Yeah? So this saves us a lot of money and gives us maybe the same amount of control and makes ambient WFI affordable for us. Yeah?
Yeah, this is really a good point. Yeah.
That's the point.
Okay, I actually have another question for Michael. Is it correct that these cutoff tests would be done by the UF supplier, not by the manufacturer?
So to be honest, the expectation here is that the manufacturer provides good and reliable cutoff data because he is the expert in the business of manufacturing a proper membrane module. And we should strictly avoid that test substances would be brought into our systems, yeah, to avoid the risk of contamination. So it should happen on the manufacturer side without touching any product that is sold on the market.
Don't do challenge tests in your water system. Yeah.
Yes.
That's the basic rule. And
Fully agreed.
so I just want to add, it's the filter manufacturer that must provide the data, yeah, and not the pharmaceutical manufacturer. And we also want to push the filter manufacturers to standardize their testing procedures, that the pharma people can compare the filters. That's our main intention. That's why we have set up this standard, this cutoff testing standard.
And this is nowadays a real mess, yeah, because everybody applies his own standard and there is no standardization. And so it comes to different cutoff values depending on the substance you apply, the pressure you apply, the crossflow you apply. Yeah, and so there are many influencing factors, and this should be really standardized. And of course, Fritz, you are right. It's not the pharmaceutical manufacturer; it's a manufacturer of the ultrafiltration modules.
Okay. And I mean, I'm sad to see this come to the end, but it looks like we are at our last question. So this question is for Fritz or Michael. Similar experience regarding ARMM water systems, not yet ready for broad implementation, cannot be the surrogate for plate count, possible regulatory concerns as well. What is the expectation regarding use of ARMMs and how are these ARMM vendors supporting?
So Fritz, shall I start?
Yeah, go on.
Yeah. So from my perspective, the regulators would like to foster further development activities. And I think someday the new technology is ready to be used as the new standard. So today it is already possible to validate ARMM against conventional technology, even for WFI. But this must be supported in a much better way by the vendors. And in addition, the measurement equipment has to be reliable and must deliver reproducible results. So this is based on our journey here with respect to ARMM. Another point is that the validation results from the vendor plus the validation results from the pharma industry should finally deliver the basis for a new limit value or for new limit values for WFI based on that new technology. Yeah.
So that's a very important point, Michael. The value that the rapid method device gives us cannot be compared with the traditional CFU. So with the incubation, yeah, we will receive different results. That's a very important topic. And so we will have to set up an action limit or a specification or whatever in the future that is not the same like the traditional 10 per 100 ml, which is written down everywhere. But anyway, I currently recommend the people to employ rapid methods because it's great for process control. You immediately get knowledge about your system, you know how it changes. I mean, currently, well, I personally know 3 manufacturers that sell microbial so online bioburden devices. Some require more invest, less OPEX, and vice versa. So depends on, and all 3 have different measurement principles. Yeah, so it's up to the user what he wants to buy. But what I see in practice is that people are not able to buy more than one RMM system as described in the Q&A. So choose the right location wisely, and we give some advice in the ISPE DACH handbook what is a good choice for a location, or yeah, but this also depends on your setup, but we are giving some tips on this in the handbook. Yeah?
So I think we all agree that that the RMM method cannot be used for quality control, for release of the product. So I think that's for sure, because it can only be used for process control. So you can check your currently, because we have no comparable limiting values at the moment. I would say when you use 3 different RMM measurements from different suppliers, they will measure completely different values. So they are not comparable in between each other. And so therefore, I think it's a good possibility to check your current microbiological situation in the system. So for example, when you have an ambient storage and distribution system, which is not continuously ozonized, you can check the continuously check the microbiological situation in your system. And if you have an increase, you may start a sanitization step. That's, for example, a good possibility to implement such a system.
Okay. Well, since that was our last question, Fritz, Michael, and Jochen, do you have any final thoughts to share with the audience just based on our conversation today?
Well, yes, I would have a short teaser for the listeners. So if you are interested in further discussions in this, so Jochen and me will give a speech together at the ISPE annual meeting Europe in Madrid next year. So join us, ask questions, and so on. If you want something more, drop us a line or visit us in Madrid, and yeah, hope to see you there.
And I think that session is on Wednesday, April 1st in Madrid. So mark your calendars and
Yes.
definitely come. It's going to be beautiful, Madrid, right? Can't go wrong, right?
Yeah, we booked good weather already for Madrid.
Hopefully. I think it'll be a really good conference, and I think we have some other things planned for our critical utilities folks. So definitely plan to come to Madrid in April. Anybody else have a plug for anything?
Nope,
Okay.
not from my side.
Okay, well, on behalf of ISPE, I would like to thank our panelists, Fritz, Michael, and Jochen, for taking time out of their busy schedules to chat with us about water. I know our water professionals are very passionate about water, so we really appreciate you sharing your thoughts with them. I know I learned a lot today, and I hope you did as well. So with that, I'll just give our quick close. I do want to thank everyone for tuning in for today's episode and invite you to come back in the future to check out our audio library, where you'll be able to search for a variety of topics important to ISPE and our members. To find the library or to learn more about us, you can visit our website at ispe.com. And if you'd like to suggest a topic you'd like to learn more about on this podcast, or even be a guest on the show to talk about something you're really passionate about, then drop us a note to see how we can get you involved. And just thank you everyone, and we'll talk again. Goodbye.
Thank you, Barbara, for being such a good moderator.
My pleasure. It was
Thank
fun.
you, Barbara. Yeah.
Thanks a
Thank
lot.
you. Bye-bye.
Bye-bye.
Bye-bye.